Hello, in this video we will discuss about PIR database, which is Protein Information Resource Database. It is mainly a mathematics resource by which you can find protein interpretations and identifications. This image shows the homepage of PIR database where you can see in About PIR tab, which is the highlighted area. You can find the history of PIR. You can simply click on history and open the history of PIR. Similarly, you can also open publications, funding and sponsors, and contact of PIR. PIR was established in 1984 by National Biomedical Research Foundation (NBRF) as a resource to assist in the identification and understanding of protein sequence information. It was also published as Atlas of Protein Sequence and Structure. The Protein Information Resource is an integrated public resource of protein informatics that supports genomic and proteomic research and scientific discovery. PIR maintains the Protein Sequence Database (PSD), which is an annotated protein database containing over 283,000 sequences covering the entire taxonomic range. In order to provide integrated and value-added protein information to the scientific community, PIR continues to enhance its three major databases, which are Protein Sequence Database (PSD), Non-Redundant Reference Sequence Database (NREF), and Integrated Protein Classification Database (IPROClass). PIR is divided into four types: PIR1, PIR2, PIR3, and PIR4. These differ in terms of quality of data. Currently, PIR1 and PIR2 account for 99% of all entries. PIR1 contains classified and annotated entries. PIR2 contains preliminary entries. PIR3 contains unverified entries, while PIR4 contains conceptual translation of sequences that are not transcribed. This picture shows the homepage of PIR. As you can see in the highlighted area, there is a search bar where you can type your protein name and search it. The searches will appear in the form of a table. And if you want to search a specific type of protein, then you should write the full name of the protein in order to search your protein of interest. Then, for example, in this image, you can see albumin protein in the search, and the searches have occurred in the form of a table. The table shows protein ID, protein name, length, organism name, Pro ID, PIR, SF ID, and UNIREF 50 ID. If you click on the protein name, a new tab will open and a general information table will appear, which will show you information like taxon group, source organism, gene name, and lineage, etc. And then as you will scroll down, the tables of cross references, family classification, features, and sequence display will be present. If you click on PRO ID, PIR, SF ID, or UNIREF 50 ID, then according to the ID type, the protein searches will appear. Next are the resources of PIR. There are 7 resources, which are IPTMNet, PRO Protein Ontology, IPROLink, IPROClass, PIR-SF, PIR-PSD, and UniProt. IPTMNet, in which I stands for integrated. It is a bioinformatics resource for integrated understanding of proteins post-translation modifications in systems biology context. Here in this picture, which is the highlighted region, this is a search bar. You can enter your protein name and restrict by organism by selecting any organism like mouse, human, rat, cow, chicken, zebrafish, fruit fly, etc., and then submit it. In the next bar, you can search the phosphorylation in literature of any protein by simply adding any keyword or protein name and then submit it. The results will look like this. For IPTM search, which is the post translation modification search, the table will show IPTM ID, protein name, gene name, organism, substrate role, enzyme role, PTM dependent PPI, sites, and isoforms. And for the phosphorylation search in literature, it will show PubMed ID, PTM enzyme, phosphorylated protein substrate, number of sentences, and text evidence. PRO Protein Ontology. PRO provides an ontological representation of protein related entities by explicitly defining them and showing the relationships between them. You can browse PRO, retrieve a PRO entry, and also search PRO by entering any text or ID. In the highlighted area, you can enter the protein name and search it. The searches will appear in this form of table, which will show you pro name, pro term definition, category, and parents. iProLink, Integrated Protein Literature, Information and Knowledge. It is a resource with access to text mining tools and annotated corpora developed in-house. Annotated corpora is the interpretations collection. iProLink consists of text mining tools. As you can see, the list is present. You can simply click on any text mining tool heading and open the tool and use it. For example, in this image, you can see iText Mine tool is open and proteins is search here. You can see PTM enzyme, substrate, site, and sentence. All the information is given. iProlink. It defines both data warehouse and hypertext navigation methods for integrating data, providing a comprehensive picture of protein properties that may lead to novel prediction and functional inference for previously uncharacterized hypothetical proteins and protein groups. In iPro class, there is a tutorial tab in the right corner. You can simply click on it and a PowerPoint presentation will be downloaded, which will guide you how to use iPro class resource. There are also options of text search, batch retrieval, ID mapping, which are the search and analysis tool. And other options are sample report, iPro class statistics and data sources, and iPro class paper. When you will click on iPro class statistics and data sources, a table will appear which will look like this. This is the data sources of iPro class. It shows protein name and ID, taxonomy, gene name, keywords, function, subunit, tissue specificity, bibliography, DNA sequence, and so on. PIR-SF. The PIR-SF concept is being used as a guiding principle to provide comprehensive and non-overlapping clustering of uniprot KB sequences into a heritable order to reflect their evolutionary relationships. In PIR-SF, you can also download the PowerPoint presentation and which will guide you how to use PIR-SF resource. There is an option of PIR-SF scan. If you click on it, then a search bar will appear where you have to enter Uniprot KB identifier and then submit it. When you will submit it, the result will appear in the form of a query sequence, which is against the set of fully curated PIR-SF families. PIR-PSD. It has been the most comprehensive and expertly curated protein sequence a database in the public domain for over 20 years. As you can see in the image, a description of this database is given, and in the highlighted area there is a search bar where you can search PIR PSD records by entering a PIR PSD ID or accession number. Then you will submit it. When you will submit it, then it will say that this PIR PSD record is no longer being maintained. It has been merged with the relevant Uniprot KB and Unipac record and is currently maintained by Uniprot Consortium. Click here to see the entry in Uniprot databases. When you will click on that link, then the searches will appear in this form of table and you can see your related information. Last is Uniprot, which is the universal protein resource, is the world's most comprehensive catalog of protein information on proteins. It is a central repository of protein sequence and function created by joining the information contained in Swissprot, Tremble, and PIR. The Uniprot has different databases which are Uniprot KB, Unimes clusters, Unipark, and Uniref. When you will click on any of these database, then a homepage of Uniprot will appear and here you can search your protein and read about what is a Uniprot. Next are the search and analysis tools, which are text search, batch retrieval, peptide match, pairwise alignment, multiple alignment, ID mapping, composition, and molecular weight. In text search, you enter text or identifiers to search against iPro class for individual proteins or PIR-SF for protein families. Here you select iPro class or PIR-SF and then enter the protein name and then search it. The searches will appear in this form. These are the results of the searches. Next is the batch retrieval. It retrieves multiple entries from IPRO class or multiple families from PIR SF database using a specific identifier or a combination of different identifiers. Here you can also select the IPRO class or PIR SF database. Then you will enter your multiple IDs in the box and then submit it. Then it will retrieve the multiple entries in this form. Here you can see the highlighted area. There is a retrieve tab. When you will click on the retrieve tab, it will retrieve the sequence. Sequence search contains peptide match, which finds an exact match for a peptide sequence, which is 3 to 30 amino acid long. It consists of two UniProt databases. Mainly, it uses two UniProt databases, which are UniProt KB and UniRef 100. UniProt KB contains functional information on proteins with accurate, consistent, and rich annotation, while UniRef 100 combines identical sequences of fragments from any organism into a single entry. In the peptide match, you can restrict by organism. You can either enter an organism from UniProt complete proteomes, or you can select organism from UniProt reference proteomes list. Then you can enter the amino acid code, and then there are the options of UniProt or SwissProt only, yes or no. Include isoforms, yes or no. Ref 100 representative sequence only, yes or no. Treat leucine isoleucine equivalent, yes or no, and then submit it. The search results will appear in this form. Next is the sequence alignment. It contains two alignments, multiple alignment and pairwise alignment. In multiple alignment, you enter multiple sequences in FASTA format or multiple UniProt KB identifiers in the ID box to get the cluster W alignment of the sequences along with a neighbor joining tree and a PIR interactive tree and alignment viewer. Here you can see there are two boxes. You can either enter the FASTA format sequence or you can enter the UniProt KB identifier and then submit it. The multiple sequence alignment will appear in this form. These sequences are multiple aligned. Pairwise alignment. In it, you have to insert two sequences using the single letter amino acid code or enter two UniProt KB identifiers. This will result in the S. Search Smith Waterman full length alignment between the two sequences. In the first box, you enter the first amino acid code or sequence, or you can enter UniProt KB identifier, and in the second box you can enter the second sequence or amino acid code and alternatively enter UniProt KB identifier and then submit it. The results will appear in this form, which are showing the full length alignment between two sequences. Other important tools contains ID mapping and molecular weight or composition. ID mapping is used to see correspondence between two identifiers and therefore facilitate the navigation among databases and analysis program. In this image you can see from ID type to ID type you have to select any two database identifiers and then enter the IDs and then map it. The results will be shown in this form, like in this picture GI number and UniProt KB identifiers are correspondence have been shown. Molecular weight and composition. These retrieve the molecular weight of a protein based on its amino acid composition. You can enter UniProt KB identifier or amino acid code and then submit it to retrieve a molecular weight. The results will appear in this form. Here you can see sequence, composition, calculation notes, all the related information is given. Acknowledgement. The PIR database is supported by grant P41 LM05978 from the National Library of Medicine, National Institutes of Health. These are the references. I hope this video helped a little bit to understand about the PIR database. Thank you.